Okay, we're going to finish up this section by talking a little bit about how Mendel's work was used to kind of connect back to cell biology and chemistry and things like that. And this took a long time, um, but it was kind of a direct result of trying to understand how inheritance worked and how inherited factors produced physical traits in living organisms and in living cells. And so this really gets us to the process of how organisms made proteins and leads us to DNA. And so the picture you see there has an E. coli, and the DNA molecule has been kind of pushed out of the cell. And so all those loops that you see is stained DNA. So pretty cool. And we have to go back to some of the stuff that we learned early on about organic molecules. These were molecules that had carbon and hydrogen bonds, and they have, because carbon can form four covalent bonds and needs to share four electrons with other atoms, it can produce really large molecules having really complex structure. Basically, you can build a molecule any shape that you want. And so organic molecules are the important functional molecules in living organisms. These organic molecules include carbohydrates, lipids, and especially proteins. And proteins are really important here. And then nucleic acids, which turned out to be also important. And the kind of really the link between Mendel and um, the cell biology really revolved around the questions of, okay, we can do this on paper. We can predict uh, the proportions of offspring of two parents if we know their physical traits or that they're true breeding. But we still didn't know what were the inherited factors that we were following, and we had no idea how the inherited factors could produce physical traits. Um, and so Mendel's work clearly worked in experimental models like the garden pea, and we could follow particular traits, but we couldn't follow every trait. But for people who are working on inherited human diseases, this really helped explain things. And this guy here, his name is Archibald Garrod, and he was working about the time Mendel's work was rediscovered. He was working on an inherited disease called alkaptonuria, which was a recessive disease and inherited just like Mendel's recessive traits um, in garden peas. And so this was a big explanation. And he had been studying this disease long enough, and he knew what was going on was that the patients were missing the ability to perform a specific chemical transformation. And they were missing that because they were missing the normal form of a particular enzyme. And enzymes are made of protein. So he proposed that these factors that Mendel was following were enzymes and that they were proteins. And at the same time, they could point to other inherited conditions that were linked to proteins. One of those was albinism, people that lacked the ability to produce a protein that was a skin pigment, melanin. And so it became through Garrod's work, it became pretty clear that the traits that um, were missing were proteins, and somehow the inherited factors affected the production of proteins. And this really made sense because proteins were involved in a whole bunch of different things. They were involved in the production of enzymes, but they were also defense molecules, transport molecules like hemoglobin. They were involved in structural support molecules like collagen and keratin. They were involved in producing motion. They were involved in the regulation of things like insulin and growth hormone. They were involved in nerve signaling. They were involved in storage and pigments. And so it was clear that whatever Mendel's factors were, the end result was protein productions that did specific jobs. Okay. Um, then the question was, what was the inherited factor? And so Mendel's inherited factors had to be moving from stem cell or uh, uh, gametes to uh, the embryo. They had to be in there, and almost right away, it was seen that these factors are probably in the nucleus, and they're moving in mitosis and meiosis. And so we could follow the chromosomes and the movement of chromosomes, and that really seem to mimic the kinds of things we would see if we were following the molecules that actually carried inheritance. And this was also true in that when we looked at pictures of chromosomes and abnormalities, we could see like, okay, Down syndrome, which is an inherited condition, is clearly associated with an abnormal number of chromosomes. Um, Other scientists did things like nucleus transfer experiments where they could take the nucleus out of a single-celled organism and put it into a different single-celled organism and take that nucleus out, and that transferred traits. And so it was clear that it was probably in the nucleus and it was probably the chromosome. Okay. And then there were only two really types of molecules in the chromosome. It was protein and DNA. Okay, and so it wasn't that wasn't that hard to figure out, and people had probably made that decision within 10 or 15 years of discovering that the proteins were the actual physical trait. And so then the question of is it DNA or is it protein has to be settled. And there were a couple really crucial experiments, and this woman here, Rosalind Franklin, um, when they determined the probable structure of protein through looking at X-ray pictures that she took. They That's when things became kind of clear, and so and that led to the models of DNA as a double-stranded ladder. Okay, the crucial experiment was probably this transformation experiment, and here it's hard to understand until you why they were doing this until you know that what they were trying to do is make a vaccine. And they were trying to make a vaccine against an infectious organism that causes pneumonia, Streptococcus pneumoniae. And when they did this, they used killed uh, disease organisms with another organism that they thought was non-disease causing, but they always produced disease in animals that were um, injected with this heat-killed uh, disease organism and live non-disease organisms. And whenever they did this, they found live disease organisms in uh, the injected animals. And they did this experiment over and over again. And it seemed somehow that inherited material information was passing from the heat-killed disease organism to the live strain that was not disease causing to change it into the disease causing organisms. And so this was a very famous experiment, and a group of people separated the chemicals in the injection and found that the transforming factor, the factor that carried the traits, was DNA and only DNA. And so this led to the model that we have now for cell function. It's called the central dogma that DNA is used to make RNA and RNA is used to make protein. I'll go through this in detail later, but this really stems from trying to figure out Mendel's work and using his system as a guideline for trying to find out what's going on in cells. So that's where we're headed in the next section, is to talk about how this happens and how we know it happens. Okay, so we're going to come back to this slide over and over again, or a slide just like it, and we're going to use that. All right, we'll stop there. I will review for the exam coming up shortly.